Hi, I'm Krista Skaggs, and I was a part of conducting a research project looking at the influence of muscle fatigue on force and EMG amplitude. Some background information on surface EMG is used to evaluate and record the electrical activity of skeletal muscle during their contraction, and shows summations of motor unit action potentials during neuromuscular activities. It's a non-invasive measure to look at variables such as fatigue and maximal voluntary contraction. Previous research has shown that motor unit firing rate either increased or remained the same as time progressed during an isometric contraction. The purpose of the study was to determine the response of motor unit recruitment during a sustained isometric contraction. I hypothesized that as muscular fatigue increased during an isometric contraction, that the force would decrease as EMG amplitude increased. The sample consisted of 8 participants, 25% female and 75% male. Their average age was about 23.38 with a standard deviation of 1.5 years. An inclusion criteria was to be a student within the Doctor of Physical Therapy biomechanics class. Some exclusion criteria were an injury to the dominant upper extremity and allergies to alcohol or adhesives, as these were used during our data collection. To collect our data, we prepped the participant's arm by shaving the area where the electrodes would be placed and cleaning it with an alcohol swab. The electrodes were placed over the wrist extensors and flexors on the dominant arm, and there was a ground placed over an inactive muscle. This can be seen in the picture on the left. We checked the placement of the electrodes and then adjusted the grip of the dynamometer to fit the participant. The participant performed a maximal voluntary contraction, which is a maximal hand grip contraction in a standardized form shown in the picture on the right. We calculated 25% of the MVC, and this is the value the participant had to maintain during the duration of the test. Peak EMG and force were recorded every 15 seconds, and the test was terminated at 300 seconds. We calculated means and standard deviations for our data. The percent standard of force was calculated by dividing the peak EMG value obtained during the standard position and multiplying by 100%. The graph of the results shows that force in gray remained constant throughout the test, while both flexors and extensors decreased over time. The trend lines for these can be seen in orange for the wrist extensors and in light blue for the wrist flexors. Our results showed that force remained constant over 300 seconds, and both the wrist extensors and flexors percent decreased over the length of the recorded test. My hypothesis was not supported. Research has shown that there can be a decrease in EMG due to a reduction in motor unit discharge rate, and the EMG may not contribute to force loss. When considering our results, the focus of exercise within the clinic may want to be directed towards concentric and eccentric training to avoid muscular fatigue.